Dennis. He always gives a over-enthusiastic, exaggerated, nice...
But not tonight, it's all true.
Yeah, he also just took me for a nice dinner, so now I'm a little sleepy. So anyways, lots of fun doing this lecture. So thank you also for coming this evening. This afternoon we had a large group, and all I could think was it was such a beautiful day. Why would anyone come during that beautiful day? But thank you for coming tonight. I'm going to tell you about the research that our program is doing, and hopefully I'll convince you that some of it is interesting. Interesting, you hear that word, interesting? I always like to start by defining what I mean by natural product, because that way we all know the same kind of definition. So nature is an incredibly, has many different habitats. It's very diverse. There are lots of different plants and animals and microbes that live in all these different habitats. And many of these organisms have the ability to make what we call natural products. They're small organic molecules that are not part of the primary metabolism of the organism. So they're not DNA or the lipids that make up cell membranes, or they're not amino acids. They're not essential to keep an animal alive, but presumably because it takes energy to make them, they make them for some reason. And so there's some advantage to that. So you've been seeing some pictures here. We're going to get to a marine picture here soon. So here is a beautiful coral reef in Curacao. And what you might notice, are there any divers in the room? Do we have any divers? Yeah? Do we have anybody who's been on glass-bottom boats or anything and seen beautiful reefs, snorkelers? On these kinds of reefs, or in many marine habitats, many of the organisms are what we call sessile. So what does that mean? That means that as larvae, they may swim around in the water column, but wherever they finally settle out and live, that's where they're probably going to live their life. There are also some mollusks that are slow-moving, so they share things that they really can't go anywhere fast. So these organisms are competing for space. They are, many of them, filter feeders. They're sponges and corals and soft corals, and then those mollusks that move slowly. And they're competing for space, and so they need to maybe make compounds that will allow them to colonize more space or keep things away. And so I always use a little analog or thing that says that if you make something that makes you like the landfill, then other things may not want to live right next to you. So you can have a bigger yard if you live right next to the landfill, because it's not a pleasant place. And so they may make compounds to do that. They may make compounds that are just plain toxic, so that if the diver or the fish come by and want to take a bite, it's bad. They're poisoned by that. They may make things that make the next generation of the fish not do very well, so the larvae of the fish don't do well. So they may make natural products to do that. They may also have situations where the very next one of their species is on a whole other reef track over. So many people who do scuba diving, there's a reef and then maybe sand and then a reef. And so if the next one of you is over there and you're going to do mass spawning with sperm and eggs into the water, if you put something into your egg that the sperm can find, you might have more successful fertilization than one that doesn't. So the compounds may have roles like that. So generally, stopping predation, stopping overgrowth, so you have more room for filter feeding, colonizing new space, modifying larval settlement, and those are all reasons why the natural products might be there. Now we just take advantage of the fact that they make them because many of the same proteins are present in these organisms as are present in us, but probably for a different reason. And so we can look to these for new medicines. So some natural products that you may have heard of in the past, you might have heard of morphine, comes from the opium poppy, used every day for painkiller. Caffeine, maybe some of you had a cup of coffee before you came here, and it comes from coffee beans. It's a natural product. The stevia-sides, some of you may have heard of the new sweetener. It's called Truvia, and there's a bunch of other names for it. It comes from a plant called stevia, and it has these very, very sweet compounds, and you can't digest those, and so you get the taste of sweet, but it is not something you get calories from. You may have heard of salicylic acid, maybe. It's found in birch trees and willow trees, and you can chew on the bark, so if you're stuck in the woods and you have a toothache, you can get some of that and chew on it, and it may work. It's been used historically for thousands of years. But you might have actually heard of this compound more, and it's called aspirin. And you see that little group, it's called an acetate to an organic chemist. And it's a very simple derivative of this natural product, which makes it so it's very stable. So you can keep that bottle of aspirin sitting in your car at 100-degree temperature and it still works because that stabilizes that structure. And the final one is just an example of an antibiotic. About 78% of all antibiotics that we use today come from nature. They're natural products. And overall, about 48% of the compounds that are clinically used that are small molecules, so not vaccines, not big biologic agents, come from nature. They're either the compound itself or inspired by nature. So the field I work in, I'm called a pharmacognosist. It's a very weird name. And we're a group of people who study natural products as medicines. And this was a big year for the American Society of Pharmacognosy because one of our members, right here, Satoshi Omura, actually is in our society, and they won the Nobel Prize. The Nobel Prize was awarded in medicine for 2 discoveries, and the first one is for ivermectins, which come from a streptomycete. And those compounds are used to treat river blindness, which is spread by sandflies. And really a partnership between the Jimmy Carter Foundation and Merck, which provides the drug for free, they're slowly eradicating this disease from the planet. And there are a number of countries now that are free of this by the use of the ivermectins and avermectins. Does anyone have a dog? Anybody have a dog here? And do you ever give it a heartworm medicine called Sentinel or HeartGuard or any of those? That's the ivermectins. And so they save the lives of a lot of dogs in the state of Florida. So they're important to me. Artemisinin comes from the wormwood plant. It's part of traditional Chinese medicine, and it's used to treat malaria. And the team that worked on that was, get the right button, Tu Yu. And it's credited with reducing deaths by malaria by 47% over the period 2000 to 2013. And so for these very important things where they're changing the lives of millions of people, they received the Nobel Prize. And so it's exciting to be a pharmacognosist and know that you can actually get the Nobel Prize for these major discoveries like this. So marine natural products. For many years, we kind of didn't have any compounds that were approved. But now there are, in fact, marine natural product and marine natural product inspired compounds that are approved. This particular tunicate can actually be found at the Fort Pierce Inlet, or at least I think it still can be, and on pier pilings in the Indian River Lagoon. And it's more common down in the Florida Keys. It lives on mangroves. And in 1967, it was first reported that if you took an extract of this organism and you put it into a leukemia model of cancer, you would get 9 out of 10 cures. That was in 67, and that was University of Miami researchers. It took till 1990 to be able to get enough compound and the right techniques to do the structure, and we were a small part of doing that structure along with a fellow named Ken Reinhardt, who's a very famous natural products chemist, who was actually my postdoctoral mentor. And it was just approved in October of this last year, so another big pharmacognosy thing for us here in Florida. It was approved for soft tissue sarcoma, and we expect that it'll also be approved for ovarian cancer fairly soon. And it's one where they can do a genetic test to see whether this particular compound will work on the tumor. So we're pleased that this has finally been approved. It took a very, very long time, 1967 to 2015, quite a long time. That's the kind of timeline that it can sometimes take. Haloven, which is shown over here, is actually a synthetic molecule, which is the right-hand side of a much larger molecule that goes across. It's not quite as complicated as that, but they found you only needed part of the structure, and it's used for metastatic breast cancer. And for many years, it was a third-line treatment. When everything else failed, then you would go to Haloven. But now they find that in some countries, we're moving to it as a first-line treatment. And this is developed by a company called Eisai, which is a Japanese company that has a big group in the United States as well. The bottom one is an interesting new kind of approach for a treatment of cancer where you have a monoclonal antibody. And what that is if you have a particular protein, you can get another protein that will recognize it and bind to it, and that's the antibody. And then so if you have a cancer cell that has a protein that's specific to the cancer, you can make the monoclonal antibody and then tie it through some spacers and linkers to what they call the warhead, and that right-hand side is what the warhead of this one is. And it's only the very end amino acid is changed from a compound called dolistatin 10, which comes from sea hares, but also probably is really made by cyanobacteria. And so this is what we call an antibody-drug conjugate, and when you treat a tumor with that, and that tumor has that, the antibody, it will selectively deliver the drug. So it overcomes a lot of the problems you have with chemotherapy from side effects. So we actually are hoping that some of our compounds that are really toxic might actually be able to be used in this kind of therapy. And in the earlier lecture, I pointed out that President Obama has said that we're going to do the moonshot for cancer, and we're going to try to make 10 years progress in 5 years. And one of the big areas they're looking at is this personalized medicine, and also trying to find where you can actually link multiple antibodies, because most tumors, solid tumors like colon cancer, breast, lung, they don't have just a single protein that's only on that cell, but they might have suites of ones that you could tie into. So I'm hoping that maybe that's the future for some of our other compounds. So at Harbor Branch, for many years, we had the Johnson C-Link submersibles, and they had a really amazing collection package that allowed us to collect unusual samples. This is actually a hexactinellid or glass sponge. This sponge is incredibly fragile, and it's growing on an absolutely vertical wall, and it would be impossible to really access this without some kind of selective submersible tool. And so we have a lot of samples like that you can't get by trawling or dredging. They really came from the collections with the Johnson C-Link. These days, we don't have the Johnson C-Links. You can actually see them. There's one on display. But we are part of the Cooperative Institute for Ocean Exploration Research and Technology. And I think you might have heard of the SEAORT, the NOAA SEAORT that's here, run by Shirley Pomponi. And what they have are remotely operated vehicles, so they're tethered vehicles that have tool packages that now allow us to go down and collect samples. And so the next little thing I have, if I can make this work properly, is courtesy of John Reed and Stephanie Farrington, and Dennis Hanasak, he's here somewhere. They are part of this Pulley Ridge project, and they said I could show this, which is showing this new Mohawk ROV and its package. And this area that's off southwest Florida, probably about 100 miles out from Sanibel and over, just north of the Florida Keys. It's about 180 feet of seawater, or 63 meters, they told me earlier. And it has an incredible diversity of organisms, including the lovely lionfish. This is kind of a little long video, but you can see some of the amazing things that are down there. This site is important because it's thought that it serves as a reservoir for fisheries through the Keys, and it also is incredibly diverse hard-bottom habitat, and it has the deepest coral that are photosynthetic, have photosynthetic symbionts. And so these are some of my favorites, the sponges and the tunicates, because those are what I like to work on. Lots of lionfish. I think they probably did scoop some. But you can see how rich it is in fish life, and that's a soft coral, and those are very important for chemistry that we work on. Dennis has any comments you can add. Lots of algae at this site. That's Dennis's specialty. Sea whip. And the ROV can operate both during the day and during the night, so they can officially, if they want, do 24-hour ops, as long as there's somebody willing to still run the ROV. There's one of the sponges that we're interested in from this area. And there's a soft coral, and one of the things to note about the soft corals, they stick off the bottom, and many, many different organisms live on them so that they can filter feed. It puts them up into the water column. So they actually serve as their own little habitat. Another lovely sponge. And earlier I thought that was a coral, but somebody told me no, it's actually a sponge, Amy. So what do I know? And that is definitely a sponge. And a thin encrusting sponge. Kind of worm. of our favorite sponges. You can see the tool package. That tool package just came online last May. It was actually built here at Harbor Branch by Bill Baxley and his team. And here's the collection of another sea fan. And we do try to make sure we don't take everything on the bottom, so we do kind of look that we're not wiping out a species. Someone asked me that earlier. We don't want to do that. Going back up to the surface, and we're going to recover the ROV. And then I now have one more, and this is just a little video of a sponge that we were really excited that they were able to recollect. It's active in one of our programs, but I'm not going to tell you which one.
This is sample number 2. It's about 15 centimeter. This is the Shrek sponge. The base is spherical, about 15 centimeters. Looks like it might have zoanthids on the surface. And then it has 2 hollow tubes coming out the top, about 10 centimeters long.
And so it's actually this particular specimen, they're not sure if it's 1 sponge, 2 sponges, or 3 sponges all growing together. They're still trying to define that. They're going to show them actually collecting it and putting it in the bucket. But this is one of our leads in one of our programs, and so getting a little bit more of it was really great that they were able to find it. And it shows that, that if we know what it looks like, sometimes we can go back and get more. So there it's going into the basket on the ROV. Oh, and I wanted to point out that the actual collection took 23 minutes, and Brian Cousin and Stephanie Farrington made that short for you. So you didn't have to spend 23 minutes watching. Imagine that. So one of the general rules of natural products work is that the more diversity of organisms that you can collect and look at, the more diversity of chemistry that you get. And you're probably not all organic chemists, but hopefully you can look at these various structures over here and get some idea that they are actually different from each other and that they may have the potential to hit different biological targets and be useful in different diseases. So we have a large collection that was collected over about 31 years. They started maybe 32 years ago in March, this March. And so we have a frozen collection, and we also have a collection of marine microorganisms. There's about 19,000 cultures of marine microorganisms from deep water sediments and organisms. So our work is, as Dennis said, really a team effort. There's a lot of different people who come together to make anything happen. So when I talk about natural products, it's really got all these different aspects to it. So we have the groups that do collections, and of course everybody likes to be able to go out and help with the collections. So once in a while we get to go. There's a natural products chemistry group, that's my group, and the people who report directly to me. There's a biological testing group, and you might recognize Esther Guzman, who speaks once in a while. She's our cancer biologist. And then this is Dee Harmody, who's doing our antibiotic testing, some of which I'll talk about later on. When we get to the point where we have an interesting compound, it's really not practical to keep going back to nature and collecting it. I mean, maybe we can farm it in aquaculture if we're really lucky, but we have to find other approaches. And so Shirley Pomponi is working on invertebrate cell culture, so actually growing the cells of sponges and things like that. We have our fermentation group because we think many of the compounds might actually be made by microbes, even if we get them from a sponge. It's a microbe making it. We've just hired a new scientist, Dr. Guozhan Wang, who is working on biosynthesis and pulling out the genes that will make the compounds. And then we're very fortunate to have good colleagues in organic synthesis who actually make the compounds in the laboratory. And most of the time, that's how things move forward to the clinic. So 3 years ago, I spoke rather extensively about how we do the chemistry, and I'm betting that that video is still somewhere out on YouTube if you really want to know a lot of detail. But I have this little brief thing where first we extract it, and when you make a cup of coffee, you're extracting coffee beans, so you use hot water. Well, we use organic solvents, but they're heated and under pressure, and that pulls out the compounds. We then do some kind of chromatography, and I think there's a little video showing this thin layer chromatography running. And you can actually see different colors going up the plate, as those are different compounds that are being separated based upon their affinity to either a solvent or to the surface, the silica gel I have on the surface there. And then finally, we do our structures by spectroscopic methods such as NMR and mass spec. And all along the way, we're testing for biological activity. So at the end of the day, my chemists end up with the compound that gives the activity that we're looking for. And there are these new immersion tours that Harbor Branch is offering. So if you don't want to go find the old YouTube thing, you can sign up for an immersion tour, and you can see our cancer biology, our microbiology, and the natural products chemistry, and you can actually spot your own thin layer plate if you really want to. I welcome you to come by on those tours. So one of the things when we make that extract, there are a lot of different compounds present. And so what you're looking at here is actually a thin layer plate with all these different compounds. These are all different species of a sponge called Batzella. And any one of these compounds might have a different biological activity than another one. So when we test the crude extract, we can have some issues. And one of those issues is that if we have one of the compounds, let's say this guy, is really toxic, but this one's useful against TB, we may not see the TB one because all the cells are dead due to the really toxic compound. So we might miss activities. And if we have really low abundance compounds, and we might not even see them on this plate, we might miss their activity as well. And there are also nuisance compounds. There are compounds that they're just active in everything, and they're not going to be useful as drugs because they're just nuisance compounds. So one of the things that we've done, and we've had funding to do, is to create this enriched library of natural products. And I talked about that a little during my last talk 3 years ago. But now I'm going to tell you some of the results from that. How do we choose what to put in the peak library? So every sample that gets collected, we do what's called an HPLC analysis. So it's a chromatography, and then we're looking at various peaks coming off. And then this is their UV spectrum. And so you can just see different peaks. These are two different organisms. They have different peaks. And this is the UV expanded. And if you look across this line 250, you can see that they're kind of shifted. And then this one has 2, and that has 2, and this one's way down here. And this is kind of a contour through the UV. And so you can see that this one especially is very rich in different chemistry. And so that would be one that we would select to take into our peak library and do the enrichment, because there's lots of different chemistry present in that. In fact, both of these are good candidates. So that's the first thing we do, is select things to go in the library. Then we actually do the chromatography using this machine. It's called a ISCO Combi Flash system, and it does a flash chromatography. And we can actually see different peaks. This is actually one of our TB leads, and you can see all these different peaks coming off. And it'll tell us what tube it put it in, and then we can dry those down. On the next slide, you can actually see one of the, a top view of the tubes, and you can clearly see there are different compounds coming off because there's, different colors that we're seeing. So those are different compounds. On the right-hand side, this is a crude extract. It's a HPLC analysis, and wherever you see one of these peaks, that's a different compound. And then these are different fractions, and you can see that this fraction, once again, is mainly one thing, and this is these guys now highly enriched. And these are these guys, highly enriched. And so once again, kind of reiterating that if we test the crude extract, and this guy is incredibly toxic, but this one was useful in Alzheimer's, we'd miss the Alzheimer's lead because we'd just kill all the cells in our assay using this guy. But now we can actually maybe see them because we've enriched them. So that's the whole goal behind this peak library, is to get to this point. So this is an example, and it comes from Esther Guzman's lab. She's our cancer biologist. You probably recognize her. She's given talks before. And this is a sponge called discodermia. And some of you who've been here many times might have remembered that we had a compound called discodermalide that went all the way into human clinical trials for cancer. It's an incredibly potent compound, much like the cancer drug Taxol. It didn't make it all the way. Every once in a while, somebody brings it back out and says, well, what about bringing it back? But it may never make it. But it's also in this extract. It's in one of the fractions that's over here. But in this particular one, Esther had two different assays. So the first one is a CCL2 assay. And so these are both in her pancreatic cancer program. And so what happens in that case is that you can get certain mutations in the pancreatic cells, and then that can cause the release of this, what's called a chemokine called CCL2. You can also have the tumor itself releasing CCL2. When it does that, it attracts to the tumor a kind of immune cell called a mast cell. So I don't know, anybody have allergies and ever have running eyes and you're sneezing and that kind of thing? If you do, it's the mast cells that are releasing agents that cause that response. So they come to the tumor and they release all these pro-inflammatory molecules, which can either drive the first tumorigenesis, so it goes from being non-tumor to being a tumor, or if it's already a tumor cell, it can make the whole tumor worse. So this compound called discodermide was in this fraction and was the active agent that blocks the production of CCL2 by the tumor cell. So we're investigating whether that might be useful in the prevention of pancreatic cancer. In IL-8 is actually a cytokine, and it's a pro-inflammatory cytokine that the cancers put out, the pancreatic tumor, and then it causes that tumor to be much more aggressive and can lead to the spread or metastasis of the tumor. And so what she found was that this adjacent fraction had a totally different compound, it's called thiopetrin K, that was able to block the production of IL-8 very selectively. And so that's another compound. We probably would not have seen, well, we wouldn't have seen either of these in the crude extract due to the presence of discodermalide. So this is just an example of how you can find things that are much more selective to treat a disease like cancer, or pancreatic cancer in this case, by doing testing of the peak library. So what are we doing with our peak library that we have made and we continue to make? We're working in malaria, tuberculosis, cancer, Alzheimer's, and antibacterial discovery. We've just started some work on ALS, or Lou Gehrig's disease, with a group at Brown University. We're doing some work in leishmaniasis, which is a parasitic disease, with a fellow named Nick Dickens, who hopefully will be here as a new faculty member before too long. And then we've been working with a fellow at UF who is going to test the library, hopefully, if we get a grant, in something called babesia, which is another parasite that's spread by ticks in the Northeast, and usually is only a problem in people who are immune suppressed, but he has a program to look for things for that. So I'm going to do a little diversion here and tell you about U.S. patent law in case you're not too familiar with patent law. We all know that we hear about patents and patent applied for. So what does a patent do? A patent gives you the exclusive right to make, to use, and to sell a product. So when you get a patent awarded, nobody else can make it, use it, or sell it without your permission. You can license it to them and they can do it, but you can, they're just not allowed to. That's what the whole idea behind a patent is. Under current patent law, we can actually patent the use of a purified natural product for, a disease, or for any use if you wanted to, as long as it's useful. And typically, if a pharmaceutical company is going to take a discovery forward and invest the up to $1 billion or $500 million to $1 billion that it's going to cost to get something approved as a medicine, they would like to see a patent on something. They would like to have some time where they can exclusively sell that medicine before it goes out into the public. There are ways, if you don't have a patent, that you can still get protection. You can do what's called a formulation patent. So you put it into a certain formulation that then no one can use that, and that might be the best way to administer the drug. So one of the tricks to patents is once you have disclosed an invention to people, you have 1 year to file a patent. So if during my talk tonight, I show you a certain compound and it's, what the structure is and the biological activity, then I have a year to file a patent. That's the law. Many of my colleagues who are the biologists that I work with are not quite ready to tell you the structure. So what they said was, I could show you biological activity for some of the compounds, but I'm not going to show you structure. And I know all the organic chemists, how many organic chemists are in the room? Lots? Yeah? So you're all bummed out. You're not going to see all the structures, but you'll see some. But this is really one of the things you have to deal with, is that you want to make sure that you have some ability to attract a company who's going to actually take it all the way. So my work is really dependent, in my opinion, to have really high impact on having really outstanding collaborators who are biologists. And so these are the group of people whose work I'm talking about tonight. And so Devapam Chakrabarty is at UCF, and he works in malaria. Esther, I already talked about her work. She's here at Harbor Branch. Peter McCarthy is here at Harbor Branch works in antibiotics and microbiology. And Kyle Rohde is at UCF at the medical school, and he works in tuberculosis. And Dr. Todd Goldie is the director of the Translational Neuroscience Institute at the University of Florida, and he works in Alzheimer's and other neurodegenerative diseases. So for each of the areas, I'm going to tell you a little bit about why we're bothering to look for something against this. So malaria. Malaria is caused by a parasite called Plasmodium. Right now, there are about 216 million clinical cases every year. About 665,000 people will die this year from malaria. 85% of those are children, mainly in Africa and Southeast Asia. There are about 3.3 billion people who live where the malaria is transmitted. And there is always, even with artemisinin, which got the Nobel Prize, there are people who are developing resistance to that. So there's always a need for some new agents for malaria. So in the United States, we don't think so much about malaria. We don't get malaria here. You have to go some other country usually to get it, and that's because we control the vector, which is the mosquito. But there's always, if you look at the CDC page, you'll find that people go overseas and they come back and they're infected, and they gets into a local population of mosquitoes, and you can still get it. But probably why our government is more interested is because we send our military overseas, and a lot of them go to places like Africa, and this is clearly a problem in places like Africa. And so, in fact, they're one of the main groups developing new agents for malaria. Our project is joint with Devapam. He took 3,000 things from our library. There were 165 that met his criteria for activity, and there were 85 different organisms they came from. And so we had to kind of winnow through those and figure out which ones we had enough funds to work on. And we've identified about 8 classes of compounds that have activity at this point. The first one is this Amphibleptula sponge, which is here. And it produces this compound called microsclerodermin A, which was originally reported to have antifungal activity and also a little activity in our pancreatic cancer models. But it's very potent, much more potent against Plasmodium, and it has about 68 nanomolar level of activity. So just to put that in perspective, you can have molar, and then you go to millimolar, which is 1,000-fold more active, and then micromolar is a million times more active, and then nanomolar is 1 billion times more active. So it's really very potent, and it's not very toxic at all to hepatocytes as a normal cell line. And so we see it has selectivity for the Plasmodium versus the normal cells of about 52-fold, which is pretty good for one of these compounds, although may not quite meet the bar that we would really like to get for a malaria drug. So I'm going to walk you through one of the things they always want to do is figure out where in the kind of life cycle or progression of the Plasmodium infection does the compound work. And so what happens is when you get infected, your red blood cell is what gets infected. And the first thing that happens after 6 hours is you get what's called the ring form. And then at 18 hours, it goes to this trophozoite form, and you can see there's something larger in there now. And then at 30 hours, it goes to the schizont form, so now there's something much larger that you can see. And at 42 hours, you go to the merozoite stage. And so what happens next is this breaks apart and releases into the bloodstream and then goes and infects other cells. So after another 6 hours, you would be back to the ring stage. So that's the normal progression that happens after the infection. So if we take microsclerodermin A, and so actually across the top are control, that's these same things here, and then at 54 hours you can see it went back to ring. If we treat at 6 hours, then it seems to progress to the trophozoite and then probably to the schizont, but then it doesn't go past. We never get the merozoite. If we treat at 18 hours, so now we're at the trophozoite stage, once again, it goes to the schizont, what's down here, but it doesn't move past. And likewise, if you treat at 30 hours, 30 hours, right here, then it goes forward into the merozoite and goes past. So if you treat, once it's already here, it'll proceed forward. So we think that it actually blocks the transition from the schizont to the merozoite. And so the next bit of work that's happening now in my colleague's lab is to figure out how exactly it does that. And so they're doing things like trying to come up with drug-resistant mutants, and then you look at the genetics of that and see what genes have changed. So that's a process that's ongoing right now, but it definitely has a very specific stage that it hits. So this is one of the compounds that they asked me not to show structure of, so we call it N1. I guess its name must start with an N. And it has about 240 nanomolar EC50 against the plasmodium and about 35-fold selectivity, so fairly reasonable. There's that same cycle again, and if we treat at 6 hours, so treating when it's in the ring stage, it really seems to kind of stay in the ring stage, or maybe it's going on to the trophozoite. If we treat once it's at the trophozoite, it goes through the whole cycle, so it continues forward. If we treat even later, once again, it goes through the merozoite and it's done. So what we think is happening with this one is that it blocks right at the, between the ring and the trophozoite. And why that might be important is that I told you about artemisinin, which got the Nobel Prize. It does the same exact thing. And interestingly, even to this day, even though that's approved as a drug and it's used, they still don't know exactly how that compound blocks this transition. And so that's one of the things in this compound that they're working on, and hopefully they'll find it out. But apparently you don't even have to know, you just have to know it does it. So, and the parasitologists tell me that the earlier in this cycle that you can hit, the better chance of it having real development potential as a drug. So I hope that that means that this will allow us to at least take it forward into the next set of studies. So antibiotic resistance. Most of us may know that there's an increasing amount of MRSA and other infections out there that you don't even have to be in a hospital to get anymore. There's a report, if you want to see it, it's out on the internet, antibiotic resistance threats. They say that if we don't do something, we'll be in a pre-antibiotic stage, so we need to find new antibiotics. You might die from a very simple infection because everything will, drugs won't work. Some statistics for MRSA are that 2 million clinical cases and 23,000 deaths every year. And you may know that if you're treated with antibiotics for a long term, you'll potentially kill all the good bacteria in your gut, and you'll end up with this guy named Clostridium difficile that will continue to grow. And it causes incredibly bad diarrhea, and it can only really be treated with a drug called vancomycin. And currently there are about 250,000 cases. Many of those people have to be hospitalized, and there are 14,000 deaths from that. And so that's when they're very worried about drug resistance in that. In terms of this cost of treating antibiotic-resistant infections, about $20 billion in direct costs for treating patients and about $35 billion for lost productivity. So finding better antibiotics, there is, in fact, some kind of need for that. And this work is done in collaboration with Peter McCarthy, and this was a sponge called Spongiostarides. And once again, the peak library fractions gave us lots of different types of activity. And this is the one here that we're focusing on that had some MRSA activity. On button. I'm a chemist, so I have to show you at least one NMR spectrum. So there's an NMR of the compound, both its proton and its carbon spectrum. And if you want to know more about that, come ask me and I'll tell you how we do structures. But we do a lot of 2D NMR, and we get a mass spectrum so we know its molecular formula. And then we put together and we end up with the final structure that we have over here. It's active at about 1 micromolar, so it's not nearly as active as some of those plasmodium hits, but it's still fairly reasonable for an antibiotic, and it also has a little antifungal activity against Candida. It interestingly has about 10-fold selectivity for the antibiotic versus normal cells, and I used to think that was not good enough, but I just found an article where it actually said that that might actually be good enough, so especially for drug resistance. So maybe there's some potential here for this compound. So the next topic is tuberculosis. I was working with, was approached by this fellow, Kyle Rohde, who took a position now, it's probably been 4 years ago or 5, at the University of Central Florida Medical School, and this was before he even went there. And he said, I heard you have this great library of things, can I test them in my TB models? And I thought, well, TB, it's not, really a big problem, is it? I mean, we read books about consumption, TB is consumption, and people used to die of that all the time, but you don't hear about people dying of TB very much, maybe a little bit. But it turns out that across the planet, it really is a problem. There's 50 million infections annually, and there'll be about 2 billion people who have what's called a latent infection. So that means that they've been exposed, and the TB is inside them, but their immune system is controlling it, and they don't have active disease. So TB typically manifests as a lung disease, but it can also hit your kidney and spleen and other things. So there's 2 billion people out there who have this infection that can become an active infection, or so if they become immune suppressed, especially in AIDS patients and things, they can have that. And then there's about 9 million new TB cases. So this many people get infected, but they don't all end up with an active infection. It's caused by a bacterium, Mycobacterium tuberculosis. It has 1.5 million deaths every year. That's where I was shocked by that, because I had no idea. Problem with current treatments is it takes 4 to 9 months. So when you get it, you have to take drugs for 4 to 9 months, and patient compliance is low. That leads to drug resistance. It's hard to take a drug for 4 to 9 months. And there were multi-drug resistant forms, about 500,000 cases in 2013, and half the people who got that died. So clearly there's some need for new medicines. So my colleague has 3 different assays and 3 different ways for looking for new treatments for TB. The first one is an in vitro assay, and that's very traditional. It's just where they grow up the bacterium, and they use what's called a microtiter plate, and it acts like a million little test tubes. And they just grow it up, and you just look for a compound that blocks it growing. They actually use a reporter gene system to detect a lack of growth. But when they did that, we had 114 hits out of 4,400, so not a high hit rate. And now there are 25 fractions that my team is currently working on, trying to find what the active compounds are. In the dormancy assay, that's that latency thing. They treat the cells, the mycobacterium in a different way, and it goes into that latent form. And then they found about 54 hits, of which 18 of those are ones that we're currently actively working on. And then the last one is a different way, where the TB will infect what's called a macrophage, which is a kind of immune cell. And in this case, these compounds that we're finding here are really only active inside that macrophage. And so the concept is that either the compounds are prodrugs, which means once they get inside the cell changes them to a new structure, and then they're active. Or alternatively, they actually activate your host cell, to kill the infection, which would be really ideal, because then you're probably not going to develop resistance. If you're actually activating the person's own immune cell to kill the infection, that's the ideal thing. So that's an area that I'm really excited about the hits they have over there, and they have quite a few. So this is just an example where we're showing that, we had a sponge and we had a fraction, and we did more separation on it, and we took it from being like 55% active, and this has actually now been diluted down, but we're getting 100%. And really, if we go to about a 1 in 100 dilution of that, we're still getting that 100%. So we've come up with a very active fraction, which turns out by NMR is still 3 closely related compounds, and we're currently working on what those structures are. We have a pretty good idea, and this is active in that dormancy assay. So far, we have 6 different compounds that we've identified, and these are ones that they don't want me to show the structure yet, because it starts that clock ticking. But we have 2 compounds in the in vitro, 2 in the latency, and 2 in the intramacrophage, and so we're continuing work on those. So I'm going to finish up by talking a little bit about our program in Alzheimer's disease. I think probably most people know that Alzheimer's is a progressive dementia that has no current cure, and it always ends in death. There are currently, in the United States alone, 5 million people who have Alzheimer's disease, and on this scale, I don't know if it's the right-hand scale, I don't know if this laser pointer is not too strong. You can see this blue line. This is the expected projection of number of people with Alzheimer's disease, about 13 to 14 million by 2050. Currently, there are 1 in 2 people over 85 have some form of dementia, and most likely Alzheimer's, but there are other forms of dementia. I think the staggering thing is that it costs us each year $220 billion to treat the 5 million people who have Alzheimer's, and about 2/3 of that is from Medicare and Medicaid. So it's a really big cost to our economy to treat this disease. You may see this little tiny yellow thing across the bottom, and that is actually the budget that's currently invested in research on Alzheimer's disease at our National Institutes of Health. It's $450 million. And to contrast that, we spend $6 billion on cancer research, and we spend $5 billion on cardiovascular health. And both of those actually have a smaller overall cost of caring for patients than that. So there is a trajectory. This doesn't show it. This is from a particular group, but we did get a bump of $300 million this year, so hopefully there'll be a little bit of investment. But when you take $450 million versus $220 billion, maybe we need to do something. Oh, and I should have said that by 2050, if we don't do anything, we're going to be spending $1 billion a year treating Alzheimer's patients. So that's a staggering number. So what are we doing? Well, we've been working with Todd Goldie, and first started working with Kevin Felsenstein. Unfortunately, Kevin has had an illness and left the university, but Todd has been part of this program from the beginning, and he took over. And we're working on trying to find things that will hopefully be used as maybe preventatives or block progression of the disease. So one of the hallmarks of Alzheimer's disease is that there are these amyloid plaques, and they're formed by a particular protein. And the way that they're formed is there's a big protein called amyloid precursor protein, and it's a transmembrane protein, and it's cut up by this protein first called beta secretase, which is a proteinase that just kind of chunks off a piece of it. Then another enzyme called gamma secretase comes along and starts cutting it into these amyloid beta peptides. And so there's a number of different ones, A beta 42, 40, 38, 37, 39. There's a whole bunch of different ones that it can cut it and release these different ones. A beta 40 is considered to be neuroprotective. So if you have a lot of A beta 40, that's good. You hopefully won't have neurotoxicity leading to the dementia. But A beta 42 has been pretty much implicated as being neurotoxic as well as forming the plaques that are one of the hallmarks of Alzheimer's disease. It's also considered that this part of the process may happen early on, and then there's another thing that happens in Alzheimer's disease where you get neurofibrillary tangles, which are made from another protein called tau, and that this probably happens first, and then you get the other, and that if you could block this, maybe the other would not progress. So the first thing that the group did was to look at gamma secretase inhibitors, and this was work by Kevin Felsenstein and others. And they took compounds. Well, the idea there is that if you inhibit this enzyme altogether and get rid of all of these, you won't get any plaques. And this was very early on in Alzheimer's disease. And what happened when they took these to clinical trials was they showed toxicity. And the reason for that, and they hadn't expected it, but it turns out that gamma secretase has other essential roles in cells. And so if you block it, those other things don't happen, and that resulted in toxicity. There also was no cognitive improvement, which then led people to say, well, maybe these plaques are not part of the disease, and maybe it's not important. But I think that if Todd were here, he'd convince you that, in fact, it is important. But that the people that you're treating in the clinical trials are so advanced in the disease that you're probably not going to reverse it. So they're not really the patient population you need to be testing. But the next thing they did, and this is where we got in our part of the project, is to look for something called modulators. So what a modulator is going to do is it's going to get rid of the bad one, the neurotoxic one. It's not going to affect other functions of gamma secretase. So all the other things that it likes to chew up, it's going to chew up. It's not going to affect A beta 40 levels or any of the others, but it's going to block the production of the plaques and hopefully the progression of the disease. And it may have to be used with the early diagnosis and then, prevent going on further. And so what they have is an assay that will let you look for compounds that can modulate the function of gamma secretase. And this is just one bit of data for some of the compounds. And if you look at this top one here, these are actually mass spectra. And so this is the A beta 37 and 38, 39, 40. That's the big neuroprotective one. And here's 42, which is the bad guy. And this is just treatment of the cells with DMSO. And then this is one of our sponge extracts. And you can see there's really no A beta 42. And this is another sponge extract. Well, it was one of our fractions. This is one fraction, another fraction, which actually you can see very little. These are really actually, as we were getting much towards more pure compounds, that I'm showing this data. And one of them you can see over here, and what they did were dose dilutions down of this guy. And even at a 1 in 4,000 dilution, which is quite dilute and probably has us down in the nanomolar potency, so once again, very potent, we're totally reducing the levels of the A beta 42. And it's actually a ratio of the good one and the bad one. So this is actually showing it's incredibly potent. So the next steps for us on this project are what we've been doing is getting enough of the compounds so that we can take them into their animal model for Alzheimer's. And for this one, we have enough, and we hope to send it next week. And this one has been really tough because when we get it really pure, it likes to decompose. And so then we cry. And there have been some tears in our lab as we've got 25 milligrams, and then it's like, oh, wait, what? It just turned black. So anyways, we're still working on stabilizing that. And we also, there is a way to make the compound synthetically now. So we think that that could be our source. So that's what we're doing in the Alzheimer's, is following up on the other active things and getting those forward for in vivo. So concluding, I hope I've maybe convinced you that our peak library is useful in identifying some interesting compounds. They may not be the compound that ends up as the drug. They may, in some cases, like even that Alzheimer's one, maybe I should go back and point at that. One of the things that we see in some of this is, see how big this A beta 37 is? According to Todd, there's no compounds known that do that. And so they're interested in that. So this may actually be a tool molecule to understand the role of A beta 37. So we have a lot more continued testing and follow-up of chemistry. We still have organisms that were the TB project. We've just barely touched what's active there. And so there's a lot more to do. And I just want to point out that this work is done by lots of different people. I stand here and present it, but there really are many, many people involved in making this happen. And so, I have to thank them. I also wanted to thank the funding agencies, and then this calls out some of the exact people who worked on each of the projects. I want to thank our Harbor Branch Foundation because they've been incredibly generous in a couple of different ways. They give the research scientists like myself what's called a Research Excellence Grant, and that has allowed me to be involved in that Alzheimer's project. It paid for all of my time to do the work on that. And also, they've been funding making more of the peak library, taking more organisms through the peak library, so we have more potential for doing more. And the Cooperative Institute has been doing the same, and our original grant for making the peak library came from NCAM and the National Cancer Institute. So with that, I'll just thank you for making it through the talk, and if you have questions, I'll try to answer them.